Yeah, welcome everyone. Today we have with us Dr. Pushpendra Singh, who is an scientist in ICMR, National Institute of Research in Tribal Health, Jabalpur. Sir completed his PhD in drug resistance mechanisms in mycobacteria under the guidance of Dr. V.M. Katoj, and then Sir started working as a postdoctoral scientist at Swiss Federal Institute of Technology. Then Sir went as visiting scientist at Louisiana State University, and then Sir was appointed as assistant professor at MS University of Baroda, and currently Sir is working as scientist E at ICMR Jabalpur. Sir's research interests include genomics and molecular epidemiology of various microbial pathogens causing leprosy, TB, scrub typhus, and different viral diseases. So, and Sir is also our research chair at Bioclues. So, I welcome you all. Thank you, sir, for taking this session. Please continue with the session.
Thank you very much, Rajul, for this kind introduction, and I thank Bioclues for giving this opportunity and this wonderful forum to share our excitement about science in some basic terminologies. And this is very exciting to be able to talk to students of schools through this type of program, bioinformatics for kids or for school children. And I see that there is a very diverse age group, and students from 5th to 12th standards are there. And of course, there are participants who are scientists and have also completed a lot of work in molecular biology and genomics. So we are very diverse group today. Considering the students, we would basically be discussing about or knowing about the diversity of life and what drives the diversity. When we see here the word life, we can differentiate living and non-living things. But something that is very common about every living thing is that they are able to reproduce and they are able to uh, make response to their environment. But what is that factor that leads to this diversity of life? How a plant is different than an animal, and then among the animals there are so much different type of life forms. There are bacteria, viruses. We all hear these things, but what is common between all these? Otherwise, it seems seems to be nothing common actually between all these. For example, in this picture, you can see so many beautiful forms of life: a tiger, a Komodo dragon, mushrooms, some insects, beautiful fishes, beautiful birds, and greenery, and so many of those flowers that basically attract our attention every day. And how they know what they are? They haven't been told. For example, recently there was a mango season, so most of you might have enjoyed mangoes, and after that you might have thrown away those seeds, goodly of mango. Sometime and in few days you would start seeing the those seeds growing. So how the mango seed that goodly knows that it has to become like a mango tree? Or for example, you might see that there are these बनियान ट्री, बरगद के पेड़ होते हैं ना, जो कहीं पर भी उग जाते हैं, बहुत छोटा सा उनका बीज होता है। वो इतना छोटा होता है कि हवा से उड़ करके कहीं पे भी जा सकता है, और पेड़ों के बीच में कहीं पे जगह मिल जाती है, या छत वगैरह के बीच में जरा सा गैप होता है, उसमें वो पानी की हेल्प से वहाँ पर ग्रो हो जाता है। आई होप आई एम अंडरस्टैंडेबल टू एवरीवन इन हिंदी ऑल्सो. If there is a concern, please write in chat box, or Rajul can tell us. But this would be basically a mix of Hindi and English whenever. So बरगद के उस छोटे से बीज को कैसे पता होता है कि उसको बरगद का पेड़ ही बनना है? एक छोटी सी एक मैंने कहीं पे एक छोटी सी कविता भी पढ़ी थी कि अ, मैं तब भी बरगद ही था जब मैं छोटा था। तो कैसे उसको पेड़ को पता होता है कि उस छोटे से बीज से ये जानकारी कहाँ छुपी होती है उसमें कि ये बरगद का ही बीज है, या उसको बरगद नीम के पेड़ से नीम ही बनना है, आम का पेड़ नहीं बनेगा। ये सब कहाँ से इतनी छोटी सी बीज में इतनी बड़ी अलग अलग, जैसे कि एक सीडी के अंदर एक अगर मूवी की एक सीडी है तो आप जिस मूवी की सीडी है वो आप प्ले करोगे तो वही वाली मूवी चलेगी ना। तो कैसे हर सीट के अंदर ऐसी क्या, हर बीज के अंदर ऐसी क्या मेमोरी रहती है, क्या इंफॉर्मेशन रहती है उसके अंदर, जिससे कि वही पेड़ बनता है उससे, या उसी से वो फूल वो फ्लावर बनता है, या हर एनिमल की इतनी जो डाइवर्सिटी है उसके पीछे क्या कारण है? इन सब में जो कि ये लिविंग फॉर्म्स हैं, इन सब की डाइवर्सिटी के जो मेन फैक्टर्स हैं वो केवल 4 लेटर्स ही हैं। पूरी इस लाइफ को अगर सिंप्लीफाइड वे में अगर हम कहना चाहें तो केवल 4 लेटर ऑफ डीएनए, 4 letters of DNA and 4 letters of RNA. They they store all this information about what should be the color of this particular flower, what would be the um, fruit, or what type of fruit this plant will bear, how long it can grow, what would be the color of this fish, and what would be its pattern on of the skin. For example, in the tiger, this type of stripes. So all this is stored in DNA. So we are going to know about DNA today, and most of these pictures you see are from the internet sources, which are informed there. So today we would be basically discussing or knowing and interacting about DNA and RNA, and then we hear about gene and genomes. These days we have heard about genomes of lot of viruses, for example, COVID virus genome. So we would also got to know about what is DNA, RNA, genome, what are the building blocks of DNA. For example, we have a building um, and it's made of bricks. So similarly, DNA is also made with certain units which are repeated, and their sequence and order determines the type of information it will contain. So we'll also get to know about the building blocks of DNA. When you perform Lego or this type of plays where you combine several pieces, so that type of pieces are there in DNA also. There are only 4 letters, 4 types of pieces, and their length, order, and how long those are. That all determines the information about DNA. And from DNA, there is RNA also produced. In some cases, RNA also can lead to the formation of DNA. So all these are known as the genetic material or the nucleic acids that have the information. Then we will also briefly know about genome size and differences in genome size in different organisms. But there are few common terminologies. For example, 12th class students may already know about the central dogma, which is basically information flow from DNA to RNA and from RNA to protein. So this is the conventional central dogma. It can be reverse also. For example, in COVID, the DNA is not present. It's actually RNA virus. So from RNA, it makes DNA by, and this is a reverse process. That is why it requires a different type of enzyme, which is reverse transcriptase. We will we have heard this terminology in the COVID time, reverse transcriptase uh, based PCR detection or RT-PCR test. So basically is from RNA to DNA, and then again from DNA to RNA and RNA to protein. So the central dogma is DNA to RNA, and from RNA the information is translated in form of proteins. So what we see is in different life forms, it's not all DNA. DNA makes RNA, and from RNA there are different types of proteins, and then the protein fold and they make their tertiary structure, quaternary structures, and that leads to so much diversity in form of an form of a color, form of in form of some structure of the material or the shape of the organism. All this is driven or manifested in form of proteins. And how then DNA replicates from one cell to another? Because if the DNA contains the information, then when one cell or one organism divides from one cell to another cell, then the DNA in every cell has to be transferred. So that is basically possible by DNA replication. So basically DNA makes photocopy of itself, and the photocopy should be just like the original ones. So it's like a color photocopy. It's not just the black and white photocopy. It has exactly the same information which is present in the original DNA, and that is by replication, by making its own copy, so that when the cell divides, both the copies of the cell have DNA exactly same. So DNA replication is one process. Another is transcription and translation. That's from DNA to RNA formation is transcription, and from RNA to protein formation is called translation. And we just now touched upon the DNA based or RNA based molecular test. For example, for COVID diagnostics, RT-PCR test was done all over the world. That was very helpful for timely detection of COVID and taking necessary preventive measures for checking its spread. Similarly, there is recombinant DNA technology, where a piece of DNA can be put into another organism so that it can make the same type of protein that we want to make from this particular gene. For example, the production of human insulin in bacteria can be done by this process of genetic engineering. This is called recombinant DNA technology. But all this is possible by knowing some basics of DNA and the cell biology. So let's see how a very simple bacterial cell looks like. It has a cell wall, cell membrane on the outer side, and inside there is a cytoplasm. So in a bacterial cell, for example, a bacteria that causes tuberculosis, it's called Mycobacterium tuberculosis, or any bacteria that generally contains a cell wall, and inside the cell wall there is DNA, and from DNA mRNA formation takes place, and from mRNA it can make protein. A protein chain is shown here in form of these beads. I hope you can follow you are able to see the cursor, Rajul. Okay. Okay. So anyway, so this beaded structure in form of like एक माला जैसा जो इसमें दिख रहा है इस पिक्चर में, that's basically the protein. So this DNA to mRNA and mRNA to protein, all this is very simplified in bacterial cell, and those are called prokaryotic cell because there is no nucleus in this particular form. Directly the DNA is present inside the cell. It can make RNA, or more precisely it's messenger RNA, because it carries message of DNA, and from messenger RNA the message again goes to protein. But in other forms, different organisms are classified into prokaryote and eukaryotes. For example, different animals and plants, those are eukaryotes, where the DNA is not present directly inside the cytoplasm, but there is a there is a nucleus inside which the DNA is present, and DNA is then transcribed into RNA, and that RNA comes out of the nucleus, and that is converted into protein by process of translation. So this is this has certain extra steps also. We are not going in details of those, but still, even in a bacteria and in our own cells, in human cells, for example, this there are some things which are still very common. The DNA has to replicate to make different type of cells, and then DNA makes its message in form of messenger RNA, and then messenger RNA is converted into proteins. So this is the standard workflow of central dogma, which is present in all life forms. There are few exceptions. We are not going in those details right now. So there are two main classes of nucleic acids. We have so far discussed about DNA and RNA. We have discussed that there are some viruses which do not have DNA, but they are RNA viruses. For example, COVID. So in those, the genetic material or the nucleic acid is in form of RNA. But again, it has to make a copy in form of DNA by reverse transcription. So DNA and RNA are generally chemically very inert. Particularly DNA is very stable. For thousands of years also it can survive, or it can remain. For example, you have seen the Jurassic Park movie where the DNA of dinosaur from some fossils was recovered. So this is actually possible from ancient bones of several thousand year old. We can detect their DNA, and by knowing their sequence of the DNA bases, we will cover that in the next slide. We are able to determine what type of features that particular organism might have, or what type of different genes it had. So while DNA is very stable, RNA is generally short-lived because it has a particular role in form of message, and it is required for a particular time. So that once that purpose is solved, that message need not to be present all the time. So it is generally short-lived. So the messenger RNA doesn't survive for very long. That is why it's present only in the living cells. And by the transcription, the RNA produces protein that we have just now discussed. So the title of this presentation was the DNA and RNA and the four letters, and how these four letters create so much diversity. So there is different combinations in which these four letters can come. So let's first know about the full form of DNA, deoxyribonucleic acid, and RNA is anyone from, if anyone can read it or read the, see in next slides why the DNA is deoxyribonucleic acid and why RNA is named as ribonucleic acid. Where is this deoxygenation take place? Where is this removal of one particular base or one particular hydrogen ion or oxygen molecule that basically gives its name as DNA. But DNA is made up of four letters: A, T, G, and C. Adenine, thymine. thymine, guanine, and cytosine. Whereas in RNA, the T is converted into U. It's replaced by uracil. Instead of thymine, it is uracil. And other three letters are same. A, G, and C are present in RNA also. But in DNA, thymine is present, whereas in in RNA it's uracil. So let's see how these look like. So since we have some very young kids also here, let's begin with the colorful picture of the basic unit of DNA, which is a DNTP, or basically it contains a nucleotide triphosphate which has a ribose sugar. That is the name that you see in the ribonucleic acid. It comes from the ribose sugar here. It's this type of pentameric structure, and the numbers are also given to these different letters. We'll come to that. And there is uracil shown here. So this is basically for RNA, because uracil is present only in RNA, not in DNA. Rajul, may I know how to basically use the cursor? Uh, okay. Yeah, pointer option. Okay, I am using arrow. Okay, yeah, thank you. I think it should be no, pointer option. Yes. Okay. Yes. Okay. Yeah, the ledger color. So okay. Yes. Ledger pen color. Okay. Yes. I ledger pointer. Okay. Now you are able to follow the. Okay. Thank you. So I was talking about this common structure that repeats in DNA and RNA. So in ribonucleic acid, you have this base attached to. So these are the when it says base, it is a nitrogenous base. A, T, G, and C. These are four type of nitrogenous bases. For RNA, it's uracil, and for DNA, it is thymine. So this base or ATGC or AUGC are attached on this particular position and on a ribose sugar. And this position is number one. And the second one is OH group or hydroxyl group. So in ribonucleic acid nucleotides or in RNA, this is OH group present on the second position of this ribose sugar. And the third position again contains another OH group, hydroxyl group. And then there is position number 4, and the position number 5 contains a phosphate group, which is shown here. So now on this position number 1 there is a nitrogenous base, and this is uracil. So since it is uracil, can anyone tell me if we are looking at the structure of DNA or RNA? If it is uracil, I think someone from the class 12th, yes, it is RNA because it is uracil. But the second clue is that this this position contains OH group, hydroxyl group, and the third position also contains hydroxyl group, and then fourth and fifth position contains a phosphate group. So this is basically a nuclear DNTP of ribonucleic acid where there are two OH groups. But in DNA, this hydroxyl group OH is replaced by a hydrogen group. So one oxygen is removed here. That is why it is deoxy ribonucleic acid. This second group contains only hydrogen. Third group contains hydroxyl OH. So because of the absence of one oxygen here, comparison in compared to the RNA, this is called deoxyribose sugar. So in DNA there is deoxyribose sugar, and in RNA it is ribose sugar. And this is important to know this numbering system 1, 2, 3, 4, 5, because DNA replication takes place in particular direction of 5 prime to 3 prime direction. So it's important to know why these are numbered differently, 1, 2, 3, 4, 5, and only on the this particular uh, 3 prime group there is another molecule added. That is why it is replication is possible only on this particular position of 3 prime, both in RNA and DNA, because it contains a hydroxyl group here. So okay, so we will be done in 5 minutes. Okay. Mhm. So in DNA and RNA, we were discussing that there is just one more difference of uracil and thymine. In thymine, there is one extra group here, which is indicated by circle, and there is absence of oxygen here. So this is DNA, and in RNA there is this extra OH group present, and the methyl group is absent here. So when you compare different pictures, you can identify the differences. In newspaper, there are some type of this type of paheli you might have seen. So these are the two differences in this picture, and that identifies DNA and RNA. So this DNA and RNA bases bind with each other in sorry these nitrogenous bases bind with each other by 3 bonds or 2 bonds. Adenine and thymine bind by double bond, and G and C, guanine and cytosine, bind by triple bond. Let's just see some colorful representation of DNA double helix. These DNA is double stranded, and you can see that different bases are binding with each other as shown in the previous slide by triple and double bonds here. And these adenine, thymine, guanine, and cytosine, their chemical structure is shown here. You can see that these have certain common features in adenine and guanine because they are of the same type. AGE are purines, and these are thymine and cytosine are pyrimidines. They are different classes. But what we mean by DNA sequencing is that when we determine what base is here, followed by this is called DNA sequencing, and that basically uh, gives us information about the genetic code. So the genetic code is triplet code, and this triplet code is in form of RNA because DNA is converted into RNA and then it is converted into protein. So when these 4 letters come in different combination and in different orientations, so they can form 64 combinations, and these two combinations form a particular type of protein block which is amino acid phenylalanine, and all these four make a single type of amino acid which makes protein, and this is a serine here. So you can see that change of the last bases are not making any changes in form of amino acid. So this is the genetic code that stores all the information, and that basically drives the diversity of life. It transfers all the information that we have in our DNA, और एक बीज के अंदर जो डीएनए होता है उसके अंदर ये एक कोड है जिसकी वजह से एक बरगद के पौधे को पता होता है कि उसे बरगद का पेड़ बनना है, आम के पेड़ को पता होता है कि आम का पेड़ बनना है, और एक मछली के बच्चे को पता होता है कि उसे उसी तरह की मछली बनना है जैसे उसके पेरेंट्स थे। बस आज के लिए मेरी तरफ से यही चीज हम कवर कर रहे हैं। जिसके अलावा डीएनए, अगर बस लास्ट स्लाइड एक और है ये कि अगर जो एक ऑर्गेनिज्म का सारा जो डीएनए है उसमें बहुत सारे जीन होते हैं, और इन कंप्लीट डीएनए सेट को जीनोम कहा जाता है। और वायरस, बैक्टीरिया, यूकेरियोट, इन सभी के अपने अपने जीनोम होते हैं। वायरसेस के जीनोम काफ़ी छोटे होते हैं, करीब 3000 से लेकर के 30,000 उसके अंदर एटीजीसी होते हैं। बैक्टीरिया में 3 मिलियन से लेकर के 5 मिलियन होते हैं, और यूकेरियोट्स में ज़्यादातर 300 मिलियन से 3000 मिलियन तक, या तीन, ये इतने बड़े जीनोम उसके अंदर पाए जाते हैं। और डीएनए अपने आप की कॉपी बनाता है जिसे डीएनए रेप्लिकेशन कहा जाता है, जिसको हम आगे कभी डिस्कस करेंगे। एक ही स्टैंड से 2 स्टैंड बनने का डीएनए रेप्लिकेशन का प्रोसीजर रहता है, वो एक पर्टिकुलर डायरेक्शन में ही रहता है, 5 प्राइम टू 3 प्राइम। इतना ही आज याद रखना काफ़ी रहेगा आपको, कि ये 5 प्राइम टू 3 प्राइम ओरिएंटेशन से ही डीएनए रेप्लिकेशन प्रोसेस चालू रहती है। ठीक है, ये मुझे लगता है कि आज बहुत सारे प्रश्न आपके मन में इस प्रेजेंटेशन से आए होंगे, और ये क्यूरियोसिटी ही एक साइंटिस्ट या साइंस को ड्राइव करती है। तो आज के लिए मैं बस इतना ही इस टॉपिक में कवर करना चाहूँगा। थैंक यू।
Yeah, we have Swarna. Do you have any questions?
No, ma'am, everything was very understanding.
Okay. And Merma, did you understand today's class?
नहीं समझना भी कोई बात नहीं है, क्योंकि ये एक आज हमने ये जाना कि सारे जो भी अलग-अलग इतनी सुंदर लाइफ फॉर्म्स जो हैं, वो सब उनके अंदर, सबके अंदर डीएनए और आरएनए से ही इतनी सारी डाइवर्सिटी, इतने सारे कलर्स, इतनी सारी शेप, इतनी सुंदर-सुंदर डिज़ाइन्स, ये सब डीएनए, आरएनए की ही कलाकारी है।
And how about you, Shivang? Did you enjoy today's class?
Yes, ma'am. Actually, Shivang just left after finishing the seminar, this presentation. He left out. I'm his mother, actually. So,
Okay,
yes,
okay.
presentation was very understanding. Thank you, sir.
Thank you, ma'am. So, okay, we have no questions. We'll continue with our hands-on session now. So, for that, we have with us today Dr. S. Anil Kumar, who is Associate Professor at the Department of Biotechnology, VFSR TR, University Guntur. Dr. Anil has obtained his master's and doctoral degree from Osmania University, Hyderabad. He completed his Serb National Postdoctoral Fellowship as Principal Investigator from JNTU, Hyderabad. And Dr. Kumar has 11 years of research and 4.5 years of teaching experience with 33 publications in various I'm sorry, in in 33 he has 33 publications in various peer-reviewed journals with cumulative impact factor of 139. And Sir has its special research interest in plant molecular biology, tissue culture, and bioinformatics techniques. He is also our outreach chair. So I'll ask Dr. Anil to please start with the presentation.
Yeah, thank you, Dr. Rajul, for introduction. Can you hear me?
Yeah, yes, sir.
Okay. Good evening to all. Myself, Dr. Anil Kumar, already introduced by Dr. Rajul. Straight away, I will move to the topic. Can I share my presentation?
Yeah.
Are you able to see my screen? Yeah. Good evening to all. Good evening to students, especially. Myself, Dr. Anil Kumar. In continuation with the previous class by Dr. Pushpendra Singh, I'll give the hands-on training on in silico gene discovery. All this time, I mean, since last half an hour or more, sir is explaining you about how DNA looks like and how DNA get converted into RNA followed by formation of proteins. In this class, I will explain you how you can discover a gene, I mean a new gene. First of all, by this time, I think you are very clear what exactly a gene is about. Gene is a segment of your chromosome that contain instructions for production of protein. There would be some areas of gene which do not code for any protein, so we are not bothered about those things for this class. All we are interested in only identifying the gene which codes for a protein. And we all know that the genes are passed from parents, great grandparents to parents, parents to your generation, from you to the next generation. And all the time, only 50% of your genome will reach to your next generation. So, in your body, you'll have 50% of genetic material or genes from your father, and the rest 50% from your mother. And the background of this story of genes started way back in the year 1800, where Gregor John Mendel, who is considered father of genetics, he started working on Pisum sativum or the garden pea plant, where he performed crosses and he came to know that from parents to next generation, some traits, at the time we call them as traits or characters, these days we are calling as characters, they are passing from one generation to another generation. And until 1900, no one approved his theory. Because of one of the three scientists, Gregor John Mendel works came into light again, and later everyone started working extensively on his works. And it was Thomas Hunt Morgan who was considered as father of modern genetics. He studied extensively on Cinderella of genetics, nothing but the fruit fly, which you commonly see on the ripened bananas. If you keep fruits aside for 2 to 3 days, you can see small flies flying on the right. Am I audible? Yeah, I made it. Is it okay? Yeah. Students, I would like to say one thing before going into the further slide. A student will get a doubts, okay, when you understand partially a concept. You're not getting any doubts after this particular class, it means only two things. One is that you have understand everything, and second, you have not understand anything. So, if you understand something, obviously you should get hello, obviously you should get doubts. So, moving on to the next slide. In the year 1900, hello sir. Yeah. Rajul, is everything fine? Yeah. In the year 1990, okay, the sequencing of human genome started. Before that, we have only very small genomes they are being sequenced. By the year 1990, they started sequencing human genomes. And by the year 2000, a rough draft of human genome has been published, which states that human genome contains around 35,000 to 40,000 genes. Later, several researchers across the globe they started working on that one, and as in today, they came down to the number count 21,000. So the human body contains 21,000 genes, 21,300 genes. So if you want to identify any new gene, you may get a doubt. What is the need to identify a gene? What is the need to study about the gene? Because if you don't know any knowledge about the gene or the genomics or the genetics, then it would be very difficult in identifying the medicines for that thing. But suppose if we keep on identifying syndromes, what is a syndrome? A syndrome is a group of diseases. The most common syndrome we always listen is about HIV, human immunovirus, which causes disease AIDS, acquired immunodeficiency syndrome. Because of this virus, we are getting a group of diseases and we are calling it a syndrome. When we are able to study what exactly happening behind the story of the genes, then only we'll come up with a medicine. If you remember Angelina Jolie, hope all of you know the Hollywood actress, right? Yes or no? Students? Hello? Guys, the class should be interactive rather than broadcasting. I'm not giving any commentary. It should be like interactive class. Hope you know Angelina Jolie, right? Yes or no?
Yes.
Hello. Yeah, fine. Good. She underwent breast removal treatment. These days, we are listening about the breast cancer frequently, okay, which is also causing death in women. When Angelina Jolie has undergone her sequencing, when she has undergone her sequencing, she came to know that she is carrying a mutant called BRCA1 breast cancer. There is a problem with one of her gene, BRCA1 gene. So she underwent breast removal and she got saved from the breast cancer disease. This is one of the simple example how sequencing or gene mining helps in understanding and how we can progress for medicine as well as development of crops. And the best another example is about golden rice. About the golden rice, especially people in Asia, mostly in China and India. The global population is represented by mostly about India and China. One third population lies in these two countries only, where they eat rice more than other things, and they're also poor nations. They cannot eat all the leafy vegetables and other fruits. So the beta carotene amount is getting lower in them. As a result, two scientists, Ingo Potricus, they had identified three genes, and they started developing golden rice. The golden rice, the name as golden rice has come because they look golden yellow in color. This is because of presence of high beta carotene. Once beta carotene goes into your body, it will get converted into vitamin A. So these are the two classical examples. One is about personalized medicine or stratified medicine by Angelina Jolie, where she has undergone treatment and she saved herself from breast cancer. And the second one is about golden rice. Now, straight away going into the hands-on experiments, I will show you the screen how you can mine the data. First of all, taking one example, for suppose you know one gene in one organism, you want to identify the same gene in other organisms, which sequencing are already done. So now I will show you hands-on experiments how to mine the gene, how to check whether the gene is the same gene whatever you had mined or not. Mining is nothing but digging the gene, and in this class, I'm going to focus on these three things: the tools, databases, and servers. The first one is the Grameen database, GenScan is a server, CDD search is a tool. So, in this class, I will show you how to work on. Please open your systems and follow exactly what I am doing. Did everyone open your system? Hello? Hello, students. Please open your systems. Okay. Hello. Swarna, Bhumika, Uzma, are you able to follow me? Hello.
Yes, sir.
Fine. Please open the NCBI database first of all. NCBI in okay. You follow me step by step, and if you're lagging at any step, please let me know. I'll stop there so that we can move together. In the Google type NCBI, right? You will get this NCBI, National Center for Biotechnological Information. Click on that one. Hope everyone is in line with me, right? Hello. Hello, Surna. Okay. No problem. I can share that one. Okay, fine. Open NCBI. In NCBI, you can see two tabs here, right? In the first one, search nucleotide. Type nucleotide. So come down, or else you can come up to letter N and choose nucleotide. Right? In the second tab, type the gene name. Type the gene name. You can work with any of the gene, but as of now, you go with this gene so that we will come up with the same results and we can discuss about the results. I chosen a particular gene from plant system, and that way I'm searching for the gene in sorghum plant. Right? Guys, are you following me or not? If there is any problem, please let yeah. If there is any problem, please let me know because at the beginning of class when I told you it should be an interactive rather than broadcasting. Okay. In the first tab, type nucleotide. We'll go very slowly. Don't worry. We'll go very slowly. In the first box, you type nucleotide, and the second one you type this one. Okay, I'm sending them through chat also. Please check your chat box. Okay, just copy it and paste it in the NCBI database and then click on the search button. You can type any gene of your interest, any gene, BRCA1, P53, whatever the gene that you're interested in, you can type. But for the experiment, I mean demonstration purpose, I will go with this gene so that you also use the same gene and we can understand the results quickly. Can you see the screen now? About students, Bhumika, Uzma, Swarna. I think most of you are attending through mobiles, right?
Yes,
If
sir.
they attend through mobile, then it would be difficult. Okay. You can take other mobile, like your father mobile or your mother mobile. You can work on these things in other mobile. You can see the screen in one mobile and you can work on the other one. Or you can open your tabs. Okay. Once once you get the sequence like this, I'm trying to find a gene that is sodium proton antiporter gene, a gene that helps in lowering the sodium in plant system. This is a gene that functions for lowering the sodium in the plant system. Okay. you simply go to the fast A. This is fast A, which is nothing but the type of the format. By clicking that one, you can get the sequence like this. If you get sequence like this, please keep it aside and in the next tab, open Grameen. In the first step, we are searching for the sequence of a known gene, okay, of a known gene, and we are trying to find out the sequence of the gene in another species. For suppose this is a sequence in sorghum bicolor, and the gene name is sodium proton antiporter. And now we are searching for sodium proton antiporter in some other plant. First, save this sequence. Open the sequence and keep it aside. In the next step, go to another database called Grameen database. Now I would like to find out this sequence in some other plant. So what we need to do is you need to blast, means you need to hit this one against all the existing sequences in the database. For that you need to click blast. Basic local alignment search tool. It is a simple search tool, something like your mobile. If you keep on typing the names whomever you wish to call, it keeps on displaying because the same way it's also show the same thing. See here. Now I'm taking this particular sequence. Okay, I'm copying the sequence. I'm keeping the sequence here in the form of FASTA format. FASTA format, all the time it should be in FASTA format because if you submit without this greater than symbol, it won't be recognized by the system, or else it will take each and every line as separate genes, which we are not interested in, which we are not interested in. So choose the gene and copy and paste. As we are working on a DNA sequences, choose the DNA and not protein because we are submitting nucleotide sequences. Go with the DNA. And now I want to check this in rice. For this, click on the change species. A new pop-up box will come like this. Type rice, nothing, type, go with the scientific name. Rice scientific name is Oryza sativa. Whether it may, you can take India group or Japonica group, no issue, you can choose anyone. Check the box. Okay, once you check Oryza sativa japonica, then check this also. Now here you can see there is a change from Arabidopsis to Oryza sativa. Take this and then we are going with a blast. Means we are hitting what are the sequences, that sequence we have taken against all the sequences that are available in the database against the sorghum sativa, Oryza sativa. Now coming back, the search tool will be blast N because we are using nucleotides and you need to take blast N only. Just click on the run option. Just click on the run option. Your result will be appeared in 10 to 15 seconds. Here you can see whatever the blast that you have submitted, it is running. Once this is running, once this run is completed, you will get the results. Wait for 32, yeah. Once the result has come, it will be shown as view results, or you can see this green in color. It has done, it has found out 57 hits, whatever for the sequence you have submitted. Just click on the view results. Yes. If you see this, hope everyone can see the page, right? Rajul, are you able to view the page? Okay, fine. Guys, very simple thing. All you need to do is you need to collect a sequence. You need to submit the sequence into the Grameen database. Grameen database contains all plant databases, all plant genomes, so that you can easily find out which species or which plant you are interested in. Once you submit the result, you will get the results like this. Okay, and here you can see the topmost showing 91% identity. Means out of the 1473, if you see this one, this is 1473. Out of this complete sequence from here to here, 91% has been matched. 91% has been matched with this particular gene. So, in order to know whether it has been matched or not, you simply click on the sequence. From this also you will get the information. Whatever the gene that you are searching for now is located on chromosome number 5. It is located on chromosome number 5, and this is the position of that particular gene. The whatever the gene you are searching for is located between this length. So click on the gene sequence. See here, you will get sequence like this. Whatever the sequence that you have submitted, it has gone and it has found a similar sequence to what you have submitted, and it is showing the result here. From this output, we can say that, okay, the submitted sequence, whatever the submitted sequence by you, is showing similarity in price on chromosome 5 at this particular position. Once this is done, okay, once this is done, you need to take the full length gene. Full length gene nothing but a coding sequence. Most of the time, your gene contains introns and exons. Exons are your coding sequences, introns are the non-coding sequences. When we talk about CDS, coding sequences, all introns will be removed, only exons will be there, and that by a process called splicing. By a process called splicing. So now I will take this particular sequence, I will get my CDS, because in the previous class I told that from DNA we'll get RNA, from RNA we'll get the protein, and from all forms of the RNA, only mRNA is able to produce protein. Okay, it can code for protein, and the rest of the RNAs they do not code for any protein. And this mRNA is nothing but your CDS. See here, the name itself is sorghum bicola, sodium proton antiporter mRNA, and that too it is complete CDS, means it's a complete coding sequences. If you look at the sequence, you can clearly see here, right? It is starting with ATG, which codes for your methionine, and if you see at the end, you can see a stop codon TGA. Any sequence will start with, I mean, any CDS, complete CDS, it will start with ATG and it ends with one of the three stop codons like TAA, TAG, and TGA. In this sequence, ATG is there at the start position, at the 5' position, and TGA is there at the 3' position. And you need to remember one more thing, that important thing, this entire length should be divisible by 3, because in the previous sessions, Pushpendra sir told that 3 codons code for 1 amino acid. So all the entire sequence, it should be divisible by 3. Now coming back to the sequencing, I mean blast result, go for configuration of this page, because we got only partial sequences. Now increase the flanking portion, means whatever the result that has come in the previous slide, I am taking upstream to, I mean to the left hand side, another 3000 bases and another 3000 bases to the right hand side, so that I can get a bigger sequence, which I will show you now. And if you remember, there are some line numbers are there, which I am not interested in, because if line numbers are there, I need to delete them manually. I don't want to waste my time and use time. So you go like this. Now you can see a bigger sequence. Can you see this? Earlier is the sequence contained not more than 10 lines. Now it is a very bigger sequence. Okay. And there are no line numbers also. There are no line numbers also. And this color portion is indicated by your exons. I told you CDS or your mRNA will be formed from your DNA. DNA is nothing but your gene. It contains both exons and introns. Once mRNA form, exons will be there. They will be removed by a process called splicing. So at the end, you will get only sorry, introns will be removed by a process called splicing. Only exons will be there. Now, if you look at this particular page, you can see presence of exons and some other elements, nothing but your introns. I will show that also. Once you get a page like this, you keep this aside and go with the another server, nothing but your gen scan. It is scanning of your gene, hence it is named as GenScan. It is a web server developed by MIT. If you open the GenScan page, it appears like this. As we are working on the plant system, choose Arabidopsis and choose the second option, that is predicted peptides and CDS. If you choose the first option, you will get only protein sequence. If you choose the second option, you will get both protein as well as DNA sequence. Whatever the sequence that has come here, simply copy that one. Simply copy that one. Copy this much big sequence and paste here. What is the main function of this particular server is, it will remove all your introns and it will give you a CDS, nothing but your only exons. Once you are done, just submit. You can see here the first sequence is around 319 bases amino acid long. Into 3 will be your nucleotide sequence, because 3 nucleotides code for 1 amino acid. Okay. Now the sequence is too short. Okay, 1960. If you come here, what are the template sequence that we have taken here is 1473 bases long. 1473 bases. And the other hand, your what are the result that we obtained right now is just 960 bases. So go to this configuration page again and configure the sequence so that you will get much more bigger sequence. I'll go with 5000 bases instead of 3000 bases. Submit the sequence. You will get much bigger sequence now. Copy the sequence and repeat the previous step again. Run gen scan. See here, now you got a much more bigger amino acid sequence. At the same time, if you see here, methionine is there at the first base. We can cross check with the nucleotide sequence also. ATG codes for methionine, and at the end, TGA is the stop codon. By this, our search is completed. We find a gene in new system, I mean new species. First of all, you had taken a gene, you had blasted against the against the species that you are interested in. After getting the sequence, this big raw sequence, we don't know how much big it is. It is close to 10,000 bases, and we had submitted that sequence to GenScan. After submitting the species sequence to GenScan, you got the result. Now the question here is, okay, if you look at the nucleotide bases, only 4 bases will be there in the DNA: adenine, thymine, cytosine, and guanine. They are repetition all through. Human genome size is around 3.4 GB. 3.4 GB contain only these 4 bases. Only 4 bases, repetition of these bases in specific patterns. So now we had discovered a minded gene in the other plant. All you need to do is, whatever the gene that you have done now is the right one or not. You need to cross check, because it's only ATCG repetition of the DNA. You may end up with a wrong data. For this reason, open NCBI CDD search. PDD search, a tool that helps in identifying whether you have mined or discovered the same gene, I mean the same gene in other plant or not. Simply go to the CDD search, conserve DNA database, submit your sequence, submit your sequence here. You can submit both nucleotide as well as in protein sequences. I'm submitting in FASTA format. Without FASTA format also you can submit, but it's better to submit in FASTA format. Click on the submit. Wait a minute, it should appear in a picture form. I think it's not showing correctly. I will take help of another tool, PFAM. For PFAM, you need to use protein sequence, not nucleotide sequence. PFAM accepts only protein sequence and not nucleotide sequence. So for the same nucleotide sequence, I am taking the protein sequence. I will submit the protein sequence in PFAM page. Sequence search. It may take a couple of minutes. Yeah, please feel free to ask questions.
Yes, ma'am. Yes, ma'am. A little bit.
Okay. What about others? Okay. Okay. The entire process is very, very simple. We are just taking a sequence. We are trying to identify whether the same sequence is in other species or not. You need to cross check whether because yeah, maybe yeah. Yeah. And we need to cross check because whether we have isolated the same one or not is very important because it's only four bases, repetition of the four base all through 3.4 GB. We need to check. How we will check? We will check whether the same domain is there or not. If the same domain is there, then we can say what are the gene that you had isolated, whatever you had bind with the help of databases is the same one. If there is no domain of that particular function, then we cannot call it as a gene. For suppose I had taken this gene sequence, sodium bipolar, sodium proton, there will be some property related to sodium functioning or transportation of sodium. If you are able to get the same result in this PFAM again, the same sodium function like thing, then you can confirm that you have mined or you have digged out a gene that is involved in sodium transport. If there is no domain in that one, then we cannot call it as a sodium gene. I'm sorry, Rajul, both the tools are down. Yeah, please note down my email ID that is there in the first slide. That is anilkumarou@gmail.com. I'll give my mail ID also. Chats. I shared my mail ID. Whoever has any doubts, please feel free to contact me. Simple. The take-home message from today's presentation is choose any gene of your interest. Any gene of your interest. Get the faster sequence and go with the other databases like Grameen and all those things. In that one, blast against your gene of interest. You will get the sequence and check whether the sequence is the right one or not with the help of GenScan server. Whatever the right sequence or wrong sequence that you submit here, it will separate the unrelated sequence and it will give you only CDS sequence so that you'll come to know what exactly the CDS is. And you submit that one to NCBA CDD search or PFAM, then it will give you the domain. Once you get the same domain, whatever the gene you are interested in, then you can call it a new mining of the same gene in different species. If not, you should not. If there is no domain, you should not call it as the same gene. For suppose, if sodium transport function is not there in the gene, whatever we have taken now from this species, if it is not there, then I should not call this particular sequence as sodium proton antiporter gene. If the domain is there, then it's okay. If not, you should not call it. This is what I try to communicate to you all. Uh, exactly, it looks like this: your Garmin database, a GenScan web server, and your CDD search tool. Just submit your sequence, you will get the domain. Once you get the domain, you can say whether it is the same gene or not. And I rest my presentation here. I will share the result once it comes. I'll share to Rajul madam. She will share the same thing to you in the group. And in the meantime, Rajul, you can share the presentation to the students so that they can cross check again whenever they are free, and they can do the homework. Okay.
Yes, sir, sure. Swarna, did you join the group with the link that I shared in the chat box?
Ma'am, can you just once again share the link because in between I left the group, so I
Yeah,
couldn't
yeah,
do
I
that.
did share it again. And hope you all enjoyed today's session. So if you have any doubts, you
Any
can
doubts,
just
please ask me. In the beginning of the class only I told you, if you're not getting any doubts, it means you have understood everything or you have not understood anything. Can I go with the second one?
I think they
Hello.
understood everything, so I'll go with the first one. Okay, thank you
Okay,
so much,
Rajul
sir.
then.
Yeah, thank you so
Yeah,
much,
thank
everyone.
you for this opportunity. Thank you, Rajul sir, and thanks to Pushpendra sir for giving pre-lecture to my session. Thank you very much.
So thank you, everyone.
Ma'am,
Yes.
ma'am,